Good afternoon, everyone, and welcome to the Georgia Tech Manufacturing Institute Spring 22 Lunch and Learn Lecture Series. My name is Dr. Billy D. Brown, and I'm a Senior Research Faculty and EWD Director at the Georgia Tech Manufacturing Institute, or GTMI. I'm excited to continue hosting this informative series along with our Strategic Partners Director, Paige Shee. I encourage you to please spread the word with our stakeholders so that with our audience, and our network, so we can provide value to our community and our stakeholders. Before I introduce today's speaker, I will first introduce GTMI and provide some brief instructions for the audience. GTMI is one of 11 Georgia Tech interdisciplinary research institutes that uniquely focuses on manufacturing research, development, and deployment. We help tackle the grand challenges of today's manufacturers and assist partners in moving innovations from the lab to the market. GTMI has a wide variety of facilities and equipment located on main campus for basic research, as well as nearby on 14th Street in our advanced manufacturing pilot facility. GTMI's mission includes education and workforce training, collaborative partnerships with industry, academia, and government, as well as thought leadership. GTMI hosts a Lunch and Learn series each semester. This spring, sessions are held Monday at noon as live online events. These sessions are excellent opportunities for Georgia Tech faculty, undergraduate and graduate students and researchers, as well as a global manufacturing community to learn and share advanced manufacturing knowledge. To ensure smooth presentation, all audience members are currently muted. If you have questions or comments for the speaker, please use the chat window or the question and answer panel. And please submit your questions as soon as you have them so they can be promptly addressed at the end of the presentation. Today, I'm pleased to introduce Professor Todd Sulczyk, who will discuss new microfluidic technologies that enable manufacturing of safer, more precise, and more cost-effective cell therapies. Dr. Sulczyk is a professor of mechanical engineering at Georgia Tech and conducts fundamental and applied research in the field of metrology, microsystems, and cell biomechanics. Dr. Sulczyk's research program creates new technologies based upon fundamentals of fluids, cell mechanical responses, and microsystem design that can manipulate cells in fundamentally new ways. The primary application include the engineering and selection of cells suitable for biomanufacturing of immune cells and stem cells for regenerative medicine and to generate new knowledge in cell mechanical responses. Dr. Sulczyk has extensive experience in translating science into technologies and technologies into products. His research has generated intellectual property and licensing agreements with both startup and established technology companies. He's a co-founder of Selfie, dedicated to bringing cell therapies to all who need them by changing how cells are manufactured, and has contributed to raising over $10 million in venture-backed funds, revenue, and grants. At Selfie, he leads the business development with pharmaceutical partners that have tested Selfie's cell engineering product. During his career, Dr. Sulczyk has received several distinctions, including the NSF Career Award, the Undergraduate Research Mentor of the Year Award, a Junior Faculty Teaching Excellence Award, the Lockheed Inspirational Young Faculty Award, 2012 Petit Institute Above and Beyond Award, Class of 1940 Course Survey Teaching Effectiveness Award, and is a Keen Faculty Innovation Fellow. At Georgia Tech, he contributes to experiential learning, particularly the teaching of entrepreneurship through CreateX, Creative Destruction Labs, and Inventure Prize. In today's session, Dr. Sulczyk will highlight the microfluidic technologies developed in his laboratory that can address challenges of cell therapy manufacturing, including cell separations, cell engineering, ex vivo culture, and fill sterility. In addition, the precise processing of cells is not enough. Microfluidic technologies must match the throughput to achieve a clinical scale. So Dr. Sulczyk will describe how hundreds of channels can be seamlessly combined to scale up throughput by order of 3 orders of magnitude to process cells at liters per minute. He will further discuss some challenges in translating technologies out of the lab and into the hands of manufacturers by relating lessons learned during validation steps. Currently, the technologies to be described are used by a number of clinical translational research labs and are being validated by several pharmaceutical companies. Dr. Sulczyk, we are pleased to have you, and you may begin your presentation.
Thank you so much, Billy D. Thank you, everybody, for joining. Uh, Let me first just introduce cell therapies to the community. They're incredibly, um, uh, have tremendous potential to help cure hard-to-treat diseases. When you think about the pharmaceutical development over the last 100 years, you're familiar probably with small molecules such as aspirin that are relatively easy to manufacture currently and relatively small size. Then, over the last several decades, protein-based therapies have become much more common. Probably the five most highest revenue-generating pharmaceutical products are all actually protein-based therapies, and that's a step up in complexity. Cell therapies are another level higher. They're a living therapy, so in potential they could stay with us for our remaining lives. And they've been shown that they can be used to cure cancer and to regenerate tissues. But manufacturing of these cell therapies is still a critical bottleneck. And if we want to make these advanced treatments accessible to all, addressing these manufacturing challenges will be a must. So the focus of this talk then will be on how we can use some new microfluidics technologies to help improve the manufacture of these cutting-edge therapies. And just to show you, these are just some photos of individuals who have gone through these cell therapies. Emily Whitehead was the very first CAR T patient through Novartis. Austin Schutz, I got a chance to meet him at one of the CMAT workshops, and it was really great. He was CAR patient number 2021. Okay, so cell therapies, they have immense growth right now. So this is a plot showing the years 2019 and 2020, the number of drugs that are being developed. These are cell therapy drugs. And if you just look at CAR T, for example, you'll see that in preclinical phase, the number is almost doubled in 1 year, with growth in phase 1, phase 2, and phase 3 as well, and as well as commercial products. These have been FDA approved and can be sold for money. Just in 2021, biotech developers who are developing cell therapies, they've raised $14 billion in funding in just the first 6 months of 2021. There's been over 20 companies that went public just in the first 8 months of 2021. And there's over 1,200 companies right now who are working on cell and gene or tissue-based treatments, and that's been an increase of over 20% in this year alone. So there's this immense growth. But then the commercial reality is that there's only a few products that are actually approved and that are actually generating revenues. Those products are listed here for cell therapies, and they're primarily in the oncology space. These are basically what's called CAR T therapies, where the CAR T cell is engineered with a CAR receptor, and that receptor is designed to bind and attack cancer cells that express that receptor. So you'll notice that the price tags associated with these cell therapies are quite eye-watering. They're quite high. And moreover, a large percentage of this price tag is actually the cost of goods and manufacturing of these cell therapies. So a quote, this is from a few years ago, but a quote was that manufacturing costs of cell therapy treatments are expected to exceed $100,000 per patient. And that's partly based on sort of the current manual processes that occur, but also just the cost of all the inputs. Okay, so if we examine the challenges, and these challenges are really, in my opinion, just becoming apparent because it's only the commercial products where you start to see the challenges become acute, but you have all this investment. So all this investment will eventually run into this roadblock of the manufacturing reality. So I would say the vast majority of all those cell products and cell therapies that are being developed are what's called autologous therapies. So these are from a patient engineered, and then back to the patient. So you can imagine it's incredibly laborious. You can't use bulk manufacturing approaches when you're making a personalized treatment. So all the inputs are quite variable and expensive. So donor cells, because you're getting this from individual patients, patients vary from donor to donor. So the inputs to the cell therapy will also vary. So, imagine if you're making a car and you have to buy your aluminum and you can't be sure of the quality of the aluminum or the constituents of the aluminum. Moreover, the inputs are expensive. So viral vector alone can cost tens of thousands of dollars, if not $50,000 or more, for high quality what's called GMP approved reagents. GMP stands for good manufacturing practices. Mentioned the personalized nature of the manufacturing. There's a large number of unit operations that are currently quite manual. It's a complex product. So there are on the order of 45 million proteins per cell. And you can imagine knowing what those are and which ones are important or correlate with good treatment outcomes is incredibly difficult to determine. And currently there's a relatively high manufacturing failure rate. With Novartis's program, they couldn't sell 10% of their products because of these not meeting the release criteria associated with the manufacturer. The manufacturing time can be extended, so 1 or 2 week culture times just to grow your cells is standard. Although that's being pushed down, and Novartis actually just came out with a 2-day manufacturing approach that I think could be quite exciting. There's logistical challenges. So right now, all the manufacturing occurs in centralized facilities. So you have to, at the point of care, obtain the cells, ship them to the manufacturing facility, and then ship them back. According to the shipping, you typically will do this with a freeze-thaw. You have to freeze your cells and then thaw them, and you can lose 50% of your cells every time you freeze them and thaw them. So again, imagine you've got a car and you want to ship a car, and if you lost, half of its power every time you had to transport it. There's safety concerns. Right now, virus is the main method for doing performing gene engineering, and virus is associated with insertional mutagenesis. And in practice, the FDA requires over 15 years of monitoring of patients that receive the virus. Some of the edits that are required, especially for sort of solid tumor cell therapies, can be quite complex. So it's not just one gene edit, but it could be multiple gene edits. And in general, it's quite difficult to determine the mechanism of action, and that leads to difficulty in determining whether your product is going to perform as you hope and as you expect. So with all those challenges, you can imagine that there's alternatives that are being sought after. An alternative to autologous cell therapy is what's called allogeneic. So with allogeneic, you can have a single donor that can then service many, many patients. So in some sense, that has great advantages. But currently, there's no commercial products, and so this isn't a reality, but it is more of a hope. But there are many companies that are developing allogeneic-based therapies. Some of the approaches, require some of these multiple gene edits, and that can lead to challenges downstream with problems such as rare events, but could be deadly, which are called chromosomal translocations. So the FDA actually halted some clinical trials associated with allogeneic companies quite recently. And then, of course, there's what's known as in vivo therapies. So these include bringing the gene editing to the cells inside the body. You don't have to remove the cells. And that can have great advantages, both logistically as well as in performing in the quality of the cells that can be engineered. And there's a tremendous amount of promising preclinical data, but again, there's no product yet, and there's going to be quite a bit of further development that's going to be needed to guarantee that it works. So for Cell therapy manufacturing, at the heart, you're genetically modifying the cells. So typically, you're providing a DNA strand that gets integrated into your therapeutic cell that can then be expanded and have a new function. For CAR T therapies, this is sort of a roadmap, simplified roadmap of what that looks like. So you have a patient, and from that patient, you extract immune cells, particularly typically T cells. Those can be isolated and then reprogrammed to generate what's called CAR T cells. Those then get expanded and grown and can be then transfused back into patient as the therapy at the point of care. So a slightly more complicated roadmap is illustrated here on the right. There's a technique called leucophoresis where you extract those immune cells. Those immune cells then get shipped to the point of manufacture, and those immune cells can then be isolated for the specific type of immune cells that you'd want, reprogrammed, expanded, and there'll be a quality check. So this is required. It's called a release criteria, and this is examining things such as sterility, proof of expression, high viability. You want the cells to be high quality. And then once the cells pass that release criteria, then they can then be re-injected back into the patient at the point of care. This figure is to illustrate sort of the diversity of cell types that can be genetically engineered that have therapeutic use. So on the top here on the left are the hematopoietic stem cells. These are often used for treatment of rare blood diseases, hemophilia, sickle cell disease, for example. Then if you look at the immune cells at the bottom row, there's cell products that are being developed both with NK cells, both the CD8 and CD4 T cells, alternative types of T cells such as gamma delta and NK T cells. And there are some products that are being developed, CAR products that are being developed for monocytes. At least 2 companies are developing monocyte CARs. On the regenerative medicine side, many of the source cells are what's called induced pluripotent stem cells. So these are typically derived from a patient from, say, dermal skin cells, transformed into stem cells, and then can be differentiated into a number of different lineages, including, for example, photoreceptor cells for treating of blindness. And then there's also adipose-derived stem cells, also called mesenchymal stem cells, which also have a lot of therapeutic promise. In terms of the payloads, this is a summary of the payloads. At the heart would be a DNA. So this might include a gene that you want to integrate into the target cell genome. You can also use RNA. So RNA is downstream of DNA. It's easier to deliver, and it's faster to get expression, but it's not going to be a permanent genetic engineering. It'll be temporary. So sometimes, for example, you can deliver your gene editing molecules that do the cutting of the genome as an RNA, and then there'll be a temporary lifespan associated with the capacity for doing cutting of the DNA. There's a variety of protein sources also that can do cutting of various types. You've probably heard of CRISPR-Cas9. There's a variety of nucleases and transposons, piggyback, TC Buster. There's a whole bunch. Every different company, it seems like, has their own proprietary brand of ability to cut DNA. And then, of course, there's virus. There's, I think, primarily lentivirus or AAV is used. And I would say it's probably the primary tool for performing genetic knock-ins, that is, delivering of a gene to a cell that doesn't exist there currently. Viruses in general have payload limits. Can't fit anything larger, any gene set larger than the carrying capacity of the virus. So that's on the order of about 8 kilobases. And I already mentioned some of the health risks. And then these inputs are then used to make the cell therapy products. I have 2 listed here in schematic form. The first is the CAR T cell. It's the CAR receptor is shown. And there's also a variety of, for example, transplanted grafts that can be developed from stem cells. This is a photoreceptor graft that can then be implanted as part of cell replacement therapies. That brings me now to my laboratory. And the vision of the laboratory is to be able to integrate many of the unit operations that are performed separately using standalone machines, is to try to replace them with microfluidic solutions. The advantage of microfluidics is that you can provide a lot of precision in the unit operation, and you also have the capacity to integrate some of these unit operations. So I got started with performing biomechanical separations. We actually wanted to make microfluidics platforms that could deform cells at very rapid time scales to study the mechanical responses, at sub-millisecond time scales. And at the time, I thought this would never be useful. I had no idea that cell separations could be an important application. But we discovered that, yeah, we could perform cell separations using microfluidics in fundamentally new ways. After that, we explored a new realm of performing cell transfections. That's where you deliver gene editing cargo into cells, and we're using mechanical forces. And so we're deforming cells at quite fast timescales that can deliver gene editing cargo in new ways. We also have a program to use sensing. So these are looking at mechanical properties of cells at very quick time frames, and that sometimes those mechanics can correlate with important functions of interest. This is an example of looking at cell viability. And in this case, viable cells are a lot softer than non-viable cells, and we can use the mechanical responses of cells as a sensor to determine whether cells are viable. And we have a recent project developing new microfluidics-enabled technologies for both sterilizations of drug products as well as inactivations of cells for allogeneic cell products. And I won't have time to talk about the last two, but I'll talk about the separations and the engineering. So I'll talk about engineering first. As I mentioned, I think the most common way of performing cell engineering is with virus. So it's either lentivirus or AAV. For GMP quality products, you're often faced with about $50,000 per patient price tag. Also, because you saw those clinical trials that are coming online, there's not capacity to manufacture a virus according to good manufacturing practices. So there's typically a long wait time. The vector size tends to be limited, and there's safety concerns. And these things all together make it very difficult to innovate in this space when you're faced with these high costs and long wait times. There are chemical permeabilization techniques. These are quite inexpensive and are often used in laboratories, but there is a need for GMP-approved reagents that can be used, and they're not always effective for all cell types. So, for example, T cells are not very suitable to liposuction. And there's also electroporation. So electroporation tools have been around for 30 or 40 years. They've been commercialized, and they have GMP-approved manufacturing capabilities. But in general, you lose quite a few of your cells. So you put your cells in a toxic media during the electroporation, and as a result, you can lose up to half of your cells just from your electroporation step. And it requires a lot of liquid handling, and so that can make it more complicated to automate. There's a number of new technologies that are coming on board now. Bruce Levine has a great review article on some of these new technologies that's listed here. They include acoustic-based methods, novel fluid-based shearing. Our approach, where we're using these ridges to deform cells, and then chemical methods that you can automate. In addition, there's a variety of nanoparticle mediated delivery, including lipid nanoparticles, and then polymeric nanoparticles as well. I think the most well-known of all the newer microfluidics approaches is a company called Squeeze, And they have a really nice system where they get all their cells to go through a single file constriction, and the cells get pulled apart when they're in the constriction, and cargo can then be delivered in through those holes. And one challenge is that larger cargo is quite difficult to deliver using this method. And so you can kind of see that in these flow cytometry plots where small cargo like 3 kilodalton can be delivered quite effectively. You can see that from the shift in the flow cytometry plot. But as your cargo gets a little bigger, in this case 40 kilodalton, which is the size of a small protein, the shift is much less pronounced. So most of the gene editing cargo is about an order of magnitude, if not 2 orders of magnitude higher in molecular weight than these cargo. Mechanical approaches, I had a fun time going back through the history books and looking at mechanical approaches throughout history. And the earliest approach I found in the literature was from 1911, so over 100 years ago, in which they're using injection to deliver gene editing cargo. But there's a number of important approaches that have been developed in the past. So we are using high-speed deformations of the cell. So the mechanical forces, and there's 2 things you need for delivery. The first is a hole in the cell, and the second is a transport mechanism. I already mentioned that many of the transport mechanisms are relied on diffusion, which just doesn't work as well for these larger cargo. We are trying to apply forces at fast time points so that we can get cells to actually lose a little bit of their volume when they get deformed. And then as they relax, they can draw in cargo from the outside, almost like a sponge. If you ever taken a sponge and squeezed it and let it go, the liquid will flow in through a convective process. So we're trying to take advantage of that for delivery of cargo. And here's a video, high-speed video, showing a process where the deformations are occurring at these ridges, which are designed in a channel, and the cells are flowing from left to right. And at each ridge, the cells are being deformed, and as they relax, the gene editing cargo can then be delivered. Every once in a while, you'll see a cell come towards an edge, and those cells tend to be swept aside. These are typically larger aggregates or other imperfections that can be pushed aside into those gutter regions. So one big advantage is that surprisingly, the cells can grow pretty well after this process. Because the deformation occurs at quite a short time point, you can actually get growth of your cells without much difference from unprocessed cells. So this in the bottom left is data from my student Tong Yu, and she's performing multiplexed editing, both with the microfluidics approach shown here, as well as with electroporation. And you can see that the electroporation growth profile is several fold different or lower than the microfluidic vecting profile that we call our technology. That leads to a great enhancement of the number of cells that you can get after a process. So that can open up new manufacturing and also shorten the manufacturing time. In general, we can process on the order of about 10 million cells per minute per channel. So if you want to go higher than that, you can include more channels. And that is basically scaling from what's called a benchtop, which typically deals with smaller numbers of cells, to clinical throughputs, where cell processing is on the order of 100 million to even 1 billion cells. To look at the physics of the cell response, I show some results from a recent paper by my student Anna Liu. Anna was the first to discover that the cells, if you deform them slow enough, they expand laterally, and so they conserve volume. But as you increase the flow rate, they no longer undergo this lateral expansion, and that we can then infer a volume change. And interestingly, there's a critical time scale that we have observed. It's about a millisecond. So somewhere on a millisecond, and it can depend on the cell biomechanics, but that seems to be the critical timescale associated with these volume loss phenomena. We've looked at this effect in a number of different cell lines as well as primary cells, and it looks like there's an interesting correlation or relationship in which the Erickson number of the cell, which is a relation between the viscous force and the elastic force of the cell, is an important parameter that can be used to predict this volume loss phenomena. And this is certainly important for us to understand more deeply and further with other cell types. Okay, so microfluidics can then be combined with different functioning agents. So this is, again, work from Tong Yu, my student, and she's integrating self-focusing steps and mechanical processing steps in a way that can be seamless. And so what she found is that she can tailor the flow profiles of the T cells flowing through from a highly focused flow profile to an unfocused flow profile. And one of the challenges that she was observed is that as that ridge gap size gets smaller and smaller, cells no longer want to go under the ridges, they want to go around. And so she observed that she was losing a large percentage of her cells with an unfocused profile, which can be simply solved by focusing. Tong has studied a number of different effects, including effective pressure. This is pressure at 3 bar, 4 bar, and 6 bar. And something I think that's interesting is that the T cell viability will decrease as you increase your shear, but delivery goes up. And so you'll have this trade-off of those two parameters. Another factor she studied is the ridge number. So this is, again, 12 ridges, 6 ridges, 12 ridges, 24 ridges, monitoring of delivery and viability. And Tong found that there's a decreasing ability to deliver. So you need at least 6 ridges in order to get good delivery. But as you go higher and higher, you lose any improvement. So when we first published this work, a number of researchers and clinicians from around the country contacted us and wanted to test the technology, and we were excited to work with them. So one of the collaborators was clinicians from Stanford, and their goal was to be able to track cell therapies inside the body. And the idea here is that if the cells migrate to the tissue of interest or the tissue where therapeutic effect is necessary, your product is more likely to work. Conversely, if your cells don't traffic to that location, then that is unlikely to work. And so instead of waiting at the end of your therapy at months, you could actually learn this in just a week or two. And so they have a workflow that they could deliver FDA-approved contrast agents to cells, but they just needed a delivery tool, a delivery method, especially one that can work in very short times inside of a surgery. So our first example, proof of principle, was with adipose derived stem cells, and the surgery works like this. You know, a patient might have adipose stem cells taken from fat deposits. The cells get loosely processed, but they're still quite a heterogeneous mixture. And then we inserted our microfluidics to see if we can deliver these MRI contrast agents, which are iron oxide nanoparticles, to the immune cells, and then at the injection to see if they trafficked to and remained in the tissue of interest. So these, for example, might be used for knee replacement surgeries. So we were very effective at delivering these iron oxide nanoparticles. We had good cell viability afterwards, and we could actually deliver about a picogram of iron to each cell on average, which was the necessary amount for MRI imaging. And in fact, we were able to get good imaging with MRI. This is in a pig, but you can see this little black dot here indicates the injection of adipose-derived stem cells and effective localization. We've since done a variety of other cell types, including CAR T cells. This is a similar experiment, but this time done with CAR T cells injected in mice. And the interesting thing is that these CAR T cells, which were localized to a tumor, a solid tumor, they could be visualized 1 week after the process and 2 weeks after the process. The cells that were not CAR T, that were just T cells, normal T cells, they did not migrate as well to the tumor. And also cells that were unlabeled also couldn't be observed, of course. This is quite exciting that we have a sort of a tool that we could use to look at mechanisms of action of CAR T therapies inside of patients over the course of weeks. Another clinician that reached out to us was in the development of stem cell therapies. This is Bud Tucker at the University of Iowa, and he has the largest collection of blindness-related induced pluripotent stem cells derived from patients. And they have a problem in that they don't have GMP approaches to manufacture and deliver these large transgenes. And so they contacted us, and we started working together, did a proof of concept that's shown here. This is a 10 kilobase plasmid that we delivered to induced pluripotent stem cells, and it has a GFP reporter, so you can actually observe whether the cells are expressing. And you can see that we're getting in all the device conditions, we're getting quite a bit better than the lipofection control that was used that's used in his lab. It turns out for photoreceptors, quite a few of them have very large transgenes, and so there's a need for new delivery technologies with microfluidics. And Bud was so excited by these results that he actually funded my lab so that we can embed one of my students into his clinical facility and his GMP and GLP manufacturing facility, so that we could give the microfluidics the best shot for integration. So with that data, we actually used my company, Selfie, to make a product for Bud. And I want to give you the workflow shown here. So Bud's lab collects these patient skin cells. From that, they make these dermal fibroblasts that then get transformed into induced pluripotent stem cells. Now, these stem cells will have the genetic mutation that causes some syndrome or some blindness, inheritable blindness disease. So in the case that was studied here, these are patients with what's called enhanced S-cone syndrome, which is one of the ramifications. It stops producing rod cells in the photoreceptor. So it's a recessive mutation in what's called the NR2E3 gene. So we wanted to see if we could perform a correction of these patient-derived stem cells, that is, supply a corrected version of that gene, knock it into the patient-derived stem cell, and then have the cell produce the correct form of the NR2E3 protein. And so so from the workflow, we perform the transfection, and then the transfection also includes it's like an antibiotic selection cassette. And from anything that survives that antibiotic has the antibiotic resistance gene, so that you can then expand and examine the clones that are hopefully NR2E3 corrected. And so we did that, and we obtained a number of clones that were successfully gene edited with what's called homology-directed repair. Our correction rate was on the order of about 1%, but that was sufficient and much better than what we got with electroporation. And you'll see that because there's two genes, two what's called alleles that are mutated, in many cases we corrected one, and in many of these successful clones we corrected one, so it has about roughly a 50% correction rate. But occasionally we could get dual corrected genes, so both alleles could be corrected such that we get 100% correct NR2E3 production. Just to see what that looks like on the genetic level, these are different reads associated with gene correction, and what the goal was to transform this what was a C into an A nucleotide. And additionally, they also, the Tucker group also mutated the PAM binding site of the Cas9 gene. So not only are you mutating the ER2E3 gene, you're also mutating the binding site of the Cas9, so the Cas9 doesn't continuously bind and edit. It'll stop the Cas9 action. And you can see about 50% of the reads here were gene corrected. In one of the clones that showed 96% corrected reads, it was beautiful. Almost every single read was gene corrected. So this is, as far as I know, this is the first time microfluidics was used for gene correction, and we think it has potential here for addressing hard to treat retinopathies in which we don't use virus. We can avoid the use of virus and obtain higher yields and also simpler processes. I mentioned that my company Selfie was the one that delivered the gene editing consumables. And I thought it would be instructive here to kind of give a quick history. So basically in 2019, my lab was being inundated with people requesting chips and devices. And so typically my students would go and make some and either mail them if they had microfluidics expertise, or travel with them to the laboratory and embed themselves if they didn't. And it was great for us to get collaborations, but it wasn't scalable. And also the making microfluidics prototypes is quite variable. These are rapid prototyping tools, but they're not consistent. And so Selfie was started in 2019 as a way of commercializing this technology. And I'll fast forward through the development cycle, but I'll just simply say that in 2020 was when we first got our venture capital funding. And then a year later, we started finishing our prototype, both instrument and consumable. And earlier versions are shown here. This is from 2021, a little bit over a year ago. We went through several development cycles, and long story short, we're able to hone in on an instrument. This is a push button instrument that can apply pressures uniformly across a multi-well cassette, a multi-well consumable, and then an injection molded consumable that has a silicon chip at the bottom of each one. And this is it's really a plug and play. Like you can literally put your cells in, put your gene editing cargo in, stick it in the instrument, press go, and 1 minute later, the cells come out engineered. This is an example of the consumable set. You can see that there's 8 wells here. Those get pressure applied. They get forced through the microfluidics channels and then pop out the other side. It's a benchtop R&D system. So it's used for basically finding the mechanical programs that are important and that work the best. And it's quite easy and quite fun to use. One thing that allows us to do is it sort of takes out the variability of the consumable and allows us to focus on the variability of the patient-derived cells or of the process. And both those things can be quite large. These are 15 patients that are processed. I'm sorry, not patients, but 15 donors, healthy donors, that have been processed with the Selfie technology. These are PBMCs delivering of mRNA, and you can see the variability of the efficiency that's primarily due to the donor, but also due to the slight variations in the process. Also, this is what's called a CRISPR-Cas9 correction. I wanted just to show you, these are 4 different bars associated with cell viability, cell recovery rate, editing efficiency, and also the product yield. And you can see that as we made these consumables with reliable silicon processing, the variability became a lot less. So these are from individual donors, but you can see that the technical replicates became a lot more precise. And now we have a clinical scale instrument that we're developing and getting ready to deliver to our customers. It's basically putting together multiple channels. In general, we can process about 1 to 10 million cells per minute per channel. So being able to process clinical scales of 100 million cells or even a billion cells is simply a matter of just increasing the number of channels. And so we've been able to do that and design a consumable set, and we can get quite fast processing. This is data of 100 million cells that were processed in 1 minute. We actually also have data of a billion cells that were processed in 1 minute, and that's quite exciting. It's some of the largest volumetric processing of microfluidics that at least I'm aware of. Okay, in the last few minutes, I wanted to present some of the separation data that we have. So we talked about the engineering, but before the engineering, we just required cell separation so that you obtain the desired cells. And the primary tools, at least in research, are what's called FACS or MACS. These are fluorescent activated cell sorting or magnetic bead activated cell sorting. And there's several limitations associated with these. The first is you need labels, and those labels should be on the surface. So anytime you use labels, you can modify the cells. And often these antibodies are derived from animals, and so you can imagine that they're not FDA approved for use in humans. You can easily modify the cells. In fact, one of the ways of activating T cells is to provide magnetic beads that have the CD3 molecule, that antibody that binds to the CD3 of the T cell. They can modify the cells by use of labels, and many cell types require multiple markers in order to identify them properly. And so the MACS and FACS are not good methods to use for those types of separations. There's also some physical separations, including elutriation, but they have a limited selection criteria. And this is just to illustrate some of the complexity of the cells that you can generate. So this is, again, from the University of Iowa, Bud Tucker's lab, who has a GLP manufacturing facility. And in the upper left, these are closed incubators that regulate the temperature and regulate the atmosphere very precisely. You can expand the fibroblasts in those through A through D. You can transform the cells in F and G to be induced pluripotent stem cells. You then fast forward 70 days and you get to J, I and J. And you can start to see that these cells, even with the best differentiation protocols, will start to produce a number of different cell lineages and cell types. These include sort of the inner and outer retina, as well as the photoreceptors that are shown in red. And then 150 days later, you start to generate your photoreceptors, and again, you have quite a big disparity of variation. Some of those cells are needed for support, and some of those cells just inhibit engraftment. And so you want to be able to select the cell types that you need. And same with immune cells. Typically, you just want one cell type. So we developed microfluidic cell sorting platforms that can sort by biomechanical properties. And it turns out that many of the cell types of interest can have distinct biomechanical properties, and that by doing these label-free sorting, we can then collect them. We figured out there's a number of parameters that we can test, and we can actually tailor the microfluidics to different cell separation modes, including stiffness, including cell viscosity, including cell size and cell adhesion. We've used this to do a number of different stem cell-based separations, including the ability to separate pluripotent stem cells from differentiated cells. That might be important to address risks of teratomas, for example. We've worked with Bud Tucker to separate photoreceptor cells. This is a complicated plot, but I'll just point out that the cell types called RPEs get directed to the first outlet, whereas the rods and cones get directed to the outlets 2 to 4. So that therefore we can enrich these retinal grafts for the photoreceptor cells and removing the cell types that we do not want. We have a number of different applications, including the purification of cells by viability. In the interest of time, I'm going to skip through these, and I'm happy to reach out to you individually so I can get to the future directions. For cell separations, the throughput's about an order of magnitude lower than the transfections. And so we're really forced to use multiplexed processing in order to generate enough cells that can be, clinically relevant. So this is our world record, at least. This is the record in my lab of processing about 60 million cells per hour in a separation output. So in this case, we've got 5 different fractions that we're taking, and we're using 2 layers of microfluidics, one to perform the separation and one to recombine all the different appropriate channels. We've also spent an effort looking at biomechanical sensing, and we can get quite accurate detection of cell viability, for example, simply by using these mechanical readouts. We've recently been funded by the NSF Future of Manufacturing. This is a collaboration with Bud Tucker. And the idea is to combine and integrate some of these cell separation platforms and transfection platforms linked together by robotic liquid handling. So his laboratory has the capacity to use image-guided selection systems in which you can remove parts of the organoid that you know to be through lots of testing, to be not to lead to good outcomes, and then to help combine and integrate separation platforms and transfection platforms using this liquid handling. So that's quite an exciting project. In future directions with CAR T cells, we have a number of clinical collaborators and research scientists that we think could really improve the processing of CAR T cells. One of those is with the processing of naive T cells. So these are T cells that are not activated, that you don't expand. The benefit is that the quality of the cells can be much improved. The problem is every single cell counts, and so you don't want to lose any. And so we're working with University of Pennsylvania in order to be able to process these naive T cells. With the complex edits for solid tumors, we're exploring new genome editing approaches, including serial editing. This allows us to cut the genome at multiple points, but separate them in time, so that you give the cell time to heal and that you decrease the risk of chromosomal translocations. I'll note that with electroporation, this is clinical data of using electroporation to do these multiplex edits. You'll see that the cell counts after an electroporation decreases by about 90%. So you can't do multiple electroporations when you're losing this much of your product. And then finally, my student is exploring the idea that you could use, because our transfection is cell mechanics based, that we can use the cell mechanics as an important input and in fact tailor the manufacturing process to either specifics of the donor or specifics of the process. And I think that donor specific or process specific manufacturing can be quite powerful to help optimize the results. And with that, I want to stop and thank everybody who helped support this. This is a picture of my students and postdocs that contributed to the project. Nick is here. He's the one that did all the eye work. He actually was hired by Bud Tucker to go and continue the microfluidics project in Bud Tucker's lab. So that project's living on. Anna did all the first transfection work, and Peter and Katie did the multiplexed cell separations projects. Here's the number of papers, and we have a number of postdoctoral trainee positions available, so please contact me if you want, and I'm happy to take any questions.
Dr. Sulchek, thank you so much for this exciting presentation around cell therapy manufacturing. I mean, there was just so much content here. I mean, it's remarkable, all the progress that your research group have made recently. It's a lot of exciting future directions as well. I want to encourage everyone, please submit any questions that you may have for Professor Sulchek. I just want to make a quick comment myself about this because that's complementary to your presentation. there's about 60 to 90% success rate with remission of acute lymphoblastic leukemia for children with this condition, this cancer, using CAR T cell therapies, which is remarkable. So I just wanted to, because I think Dr. Sulchek, you did a great job of just actually, looking at a lot of the imperfections and the challenges involved with the manufacturing of it. But I think some of the uh, that's really the motivating factor, though, is the medical right success. I'm sure you're well aware of that. The
Yeah,
success of these
absolutely.
CAR T cell therapies, exactly,
Yeah,
is
absolutely, Billy D. And
very exciting and very motivational.
yeah, I agree. And a lot of these treatments they're used sort of as the last resort. Um,
Right.
as we improved our manufacturing and our ability to scale these cell therapies, it might be possible to use them earlier on, maybe as a first resort. And I would anticipate that the success rate might increase if you could address some of these patients at an earlier time point.
Exactly. And I'm sure also bringing down the cost of the therapies will help as well to create more
Yeah,
clinical outcomes.
absolutely. As I mentioned, if you're a small startup and it costs $100,000 to buy a virus to your 20 patients for your clinical trials, it's going to be very hard to innovate in that space.
Yeah, actually, I wanted to ask about that. Do you mind just quickly summarizing, the for the cell engineering, using viruses versus not using viruses with your technology, for instance, with Selfie?
Yeah,
What are some of
absolutely.
the
So,
advantages and disadvantages of using viruses or not for
right.
trans? Yeah.
So, as far as I know, for clinical products, the only time electroporation, which is the um, main manufacturing tool besides virus, is with what's called genetic knockouts. So these are when you can disable a gene by cutting it using, say, CRISPR-Cas9. The more important, maybe more important application is delivering a gene and inserting a gene. And that can require a Cas9 cut, but you also need to supply the DNA template. And viruses are exceptional at being able to do this efficiently, but a non-viral approach that could do this would be immense. I've seen great data, preclinical data from a number of groups in the CMAT consortium, including Chris Saha had great transfection efficiency and knock-in efficiency with homology-directed repair. So I'm excited to see that develop. I think microfluidics might provide an alternative, especially if you need cells to be able to expand and recover after your process. Our stem cell work is the first time I've seen this homology-directed repair and knock-in editing done with microfluidics. So I'm really excited to see that work be published.
Great, great. I have a question here, actually from Reginald Tran. The clinical scale up is very impressive. How about the opposite end with small scale pilot transfections for rare cells or small patient samples? Is there a minimum
Yeah,
number of cells for screening and optimization purposes?
great. Thank you, Reggie, for asking about that. We get asked about this all the time. That be able to just can you screen small volumes and small numbers of cells? And in principle, you think microfluidics is the best in the world at this. In practice, with liquid handling and at least our current product, we can handle about 50 microliters of cells. If you go, that's kind of our sort of our dead volume, so to speak. So that I think that sets the limit. And even at 50 microliters, that's not very reliable. I mean, it'll be you might lose or not get any results at all. A third of the time or a quarter of the time. So on the small end, if you think about a million cells per mil, you're looking at less than 100 microliters. I think we can reliably process 100,000 cells. Going much beyond 50,000 is a challenge currently. But I think with improved liquid handling, smaller volumes, and just more experience, we can push those smaller volumes down even further.
All right. Well, we're actually at the end of the hour, 1 o'clock, so I don't think we're going to take any more questions. Um, Dr. Sulchek, I just want to thank you once more for your time, and this has been very valuable, particularly even for me, kind of because I've been working on a project funded by Army BioFab USA for sensors and monitoring of bioprocesses. And so, my biological expertise has been growing, but I think that this is very helpful for me. So I definitely want to keep in touch with you, and I just want to remind the audience that the content shown today it is recorded and it will be posted. A link will be posted on our website, so you can view the recording or potentially share it with others. So thank you, Dr. Sulchek, again for the presentation, and I want to thank our audience for joining. I do want to remind you next week we'll have another seminar this will be from Matthew Jackson, who's the Director of Strategic Technology at Solvay. So he'll be talking about composite applications, manufacturing, and the vision forward. So thanks, everyone, and have a great Monday.